Thank you, Dimitri. So I apologize for some of the slides. I couldn't download. Some of my talk didn't work. And I downloaded the ones, and those are not the most recent one. The one thing that I want to discuss, we hope that we can maintain this date. Currently we have a problem to find enough rooms for that. Okay, let me get into where we were in that talk. Oh. So let me begin with this one again. When we begin with a protein sequence, what's the first step we do with it? Has that become clear? I'm going to make more points about that. I mean, in particular for the people who did not take last semester's course. You start with the sequence. What do you do? People who so the qualified for answers are only people who have not taken the last semester's course. Have I sort of made that? Why do you smile?
I agree with him, like, depending on what you want to do. It depends on what you want to do with the...
Oh, that's what he did. Is that what he said?
Yeah.
Okay. So what could you, for instance, want to do?
Compare it with something else.
What would you compare it to? I mean, what can you see here that reminds you of something?
An alignment with other...
Does everybody know what that means in alignment? When you sort of roughly know, can anybody, again, I'd like answers from people who have not taken last semester's course.
You try to find a match with another one, the one that knows.
Exactly. So you would, in this particular case, just look at the first three letters here, and you immediately see the first three letters are already identical in both sequences. And you could also look at the name. Then you immediately see that there's some similarity between these two. But the way you do an alignment simply is you take this sequence, so the string of letters that you have, and compare them one by one. And again, somebody who has not taken last semester's course, how would you do that? Any idea? Any proposal? What would be the simplest way to do? So one way to optimize really is that you optimize the number of identical letters, right? So I remember I told you that proteins consist of units. So it may, in this particular case, so visually you immediately see those have equal length, and something immediately somehow fits on the screen. And since they are chosen to be the same name, it's clear that they are related. But when you begin with that sequence, you may not know. So you may also look for sort of substring matches. You may look for things that are much shorter than this entire thing and see whether this much shorter thing, often referred to, for instance, as a domain or a functional unit, is visible here. So how would you do that? Simplest way. The simplest way is you do the random superposition, right? And then you count. And you do that many times until you find something that is better. Does that strike you like a good idea? Why not?
It's too many combinations.
Takes too long. So one thing is already clear. We cannot quite... well, actually, let's turn that around. Let's say we have 100 residues in each. Can we do all against all? Can we just do all combinations? In this particular case? 100 times 100? Easy, right? We can do that. The story gets a little bit more complicated if you run this against 50 million as we have it in the database. That's where the trick becomes complicated. But in principle, we could do a completely brute force all against all. And that, in fact, is the simplest way to do alignments. You simply slide, so you begin with some sentence or some part of that, and you slide it through and see how well it fits. It's a dynamic programming algorithm that matches it to something that is relatively easy to do, but it's still square in the length. So it's still of the order of the product of the length of the two. it is in fact taking a lot of time, which does easily work for 100 by 100, but for 100 by 100 times 50 million it begins to become a problem. And then there are fast hashing solutions that do this much faster. And again, I'm not really going to talk about it, but this is always the most important thing that people start with. I mentioned that function is known for about 10 to 50% of all the human proteins, and the spread of these numbers has to do what exactly you define as function, how much resolution you want to have in order to say that the function is known. In principle, the problem is sort of a trivial one. Epstein and Anfinsen showed experimentally that sequence uniquely determines structure. That means you take a sequence, the one that I showed you, put it into a solvent, it adopts a unique three-dimensional structure. Unique means you do it 10 times, gets 10 times the same. That unique three-dimensional structure is exactly relevant for the way it functions. So all the information essentially is in the sequence and in the solvent, the interaction between sequence and solvent. That, however, no longer is an easy to solve problem. So the space of the dynamic space of a protein structure formation is big. Now, it's not only that the prediction of protein structure is difficult from sequence. There's another aspect here, captured by molecular dynamics, and in fact protein function is motion. What I mean is that if you imagine two bricks meeting each other and docking, there must be cases in life, in our daily experience, where something like that happens, right? Like two things that come together and do something, I don't know. But the world of biology is not like that. And the point is that the action, it is the idea of key and lock. So it's always the background, in particular for drug design and things like that. There's very often this background that a drug fits to a protein if key fits to a hole. But the thing where this analogy completely breaks down is that upon inserting the key into the hole, you change the hole. Or the key, or both. So there is motion, okay? There is, in fact, in principle, everything in chemistry is like that. It's not the world of stones of physics. So the interaction is a probability, and it happens in a dynamic sense. That means you fit better or worse. Let me just show you some movies that are supposed to show this. And that may all be a victim of my wrong computer. Yes, bless you. You see this brilliant motion here. Okay, this is very difficult to see because of the resolution. The PDF didn't download the movie, and my talk, but wait a minute, the talk should have the movie embedded in it, right? Find it. It's not here. I put it. Presentations is in. Through the entire hierarchy? You dare a lot. I don't dare these things. And on top it's not my computer, so I know nothing about it. It's full of old stuff. Oh, why is that?
Maybe you have it somewhere online.
I have somewhere what?
Online.
Online?
Online.
Ah, no, that's the problem. So I just downloaded the online talk and that doesn't have the movies. The PDF, somehow the PDF didn't take the movies. Okay, I'm going to give that next time to you. There's no point that I waste more time on this now. Next.
Please continue.
Please continue wasting time. Okay, so I'll get back to this idea of motion is it, and the way, this is a short excerpt from David Shaw's talk. We get back to that on Tuesday. Now, let's turn into a different part. So this semester is about predicting function, and predicting function always has to do with an objective function, so function has to be defined somehow. When last semester we talked about comparing sequences, so that's the alignment. One objective function I immediately gave you now, which is that we match identity of letters. It's a simple criterion that you can optimize. It's a simple way to measure the similarity between two proteins, that you simply count how many letters are identical. There are other ways to measure that. In order to compare to structures, typically what you do is you do an RMSD. So when you have these two objects, you want to superpose them, you find corresponding points, you turn it around in this particular case, and then you measure in this space here how close are the corresponding fingers to each other. And the simplest way would be a Hamming distance. So a root mean square distance between all the points that we have, right? And then you can simply optimize a method that predicts, for instance, structure. You have a structure that you experimentally observe, and you measure, you have a structure that you predict, and then you simply measure the similarity between the two. What about function? In function, this is more complicated, because function is not anything that I can measure in 3D. It's not anything that I can easily write down in a number. Here is one attempt that tries to, in fact, map it to a number, and I will bring this back again in next week. This is the enzyme nomenclature. And what is important to carry home from the lecture, I believe, is that function is nothing that we can write into a simple number, and that there are several attempts. The two major attempts, I will present them in more detail, so this is the first time I present them, and that's here the EC number. The EC number has, on the highest level, the so-called first number, 6 different numbers. And these 6 different numbers have to do with the chemistry it does. So ligases, they bring things together. Hydrolases, transferases move things, reductases cut things. So it is essentially about the type or the kind of action in the chemistry sense that the enzyme does. Okay? It's like you classify your tools at home. And that gives you the first number. So EC1 means the first digit in this particular is an oxyreductase, then the first digit would be 1. If it were a ligase, then the first digit would be 6. Okay? Then we get further down, if we are talking about a lyase, the next issue is, what is the substrate? What is it bringing together? Here is carbon-carbon, here's carbon-oxygen, and so forth, and so on. These again, so simply you go through the chemistry that is possible, or that you believe. Chemists go through the chemistry that they believe, or biochemists that they believe is possible, and then they name all these reactions that they have seen and give out numbers. Okay? Then you go to further detail, and further detail. The way this is organized is strictly such that when you go further down in the so first of all, we have always 4 levels. Okay? There's no other enzyme. Enzymes are either classified or not. If they are classified, they are put into these numbers. Secondly, there's a 1-to-1. So maybe your enzyme has some features of that and some features of the other. It may get 2 classification numbers, but it has this essentially one number for one feature. And the 2 classification numbers typically come from 2 different regions in the protein. Typically, the assumption is that one region does one thing, has one number. Okay? There's a 1-to-1 in that sense. Whether you are an EC1 in oxyreductase or in the ligase in the other side here, deeper, the third level, the second level is sort of comparable in terms of the level of detail you provide. Second level is always the type of substrate. Then they deviate a little bit because the reactions become different. But you still, where you can compare it, level 4 for all classes here is the deepest. Yeah?
How many rules does this nomenclature have?
I'm not entirely sure what you mean by rules. So how many classifications? 4 steps.
Is it a tree or a forest?
It's a tree.
It's just a tree, so it has 1 root.
Oh, so many roots. Oh, I'm sorry if I understood rules. One.
And what is it?
The enzyme.
Just the enzyme.
So it is an enzyme. If it is an enzyme, then the question is what type of these 6 enzymes it is, yes?
I maybe didn't hear it. We only talk about leaves in this case. Or
Yes.
is there also the possibility of an enzyme being in the middle layer?
No.
Okay.
We have leaves. So each enzyme, the enzyme may not be assigned the last digit because they may not have done the experiment that completely shows which leaf it belongs to, but the underlying philosophy or assumption is that there is one enzyme for one leaf, or one functional unit for one leaf, yes?
And all leaves are only on the 4th letter.
Yes, correct. All of these things are true, and this is makes the hierarchy very strict, very simple. Very simple for computer scientists to deal with. Unfortunately, the simpler, this is sort of a common theme in biology, the simpler the nomenclature for us is, the more removed it is from reality.
How do you name the complexes, enzyme complexes or something? I mean,
Yeah, so essentially you say one unit has this and the other one has that, and you give both numbers.
like more active sites and stuff.
And you give 2 numbers, exactly. And then, so again, today, I'm not entirely sure, until very recently, most of these assignments were not done in a way where people exactly knew where the enzymatic activity is. For most assignments in the database, they had an idea, so they saw the reaction. How the protein does that reaction, they didn't know. So in most of the cases, when you give 2 numbers, you do not know whether these domains are actually different. This is what we assume. Typically that is the case. But for most of the cases, we don't have this evidence. In some cases we do. In some cases we actually have evidence that there are similar regions. And similar regions, again, what you said fits to that idea. So you have 2 different active sites. So essentially you have 2 sides of it's a single domain, but you have 2 different sides of the molecule and they can do slightly different things. Right? Okay, the point is on the sequence, they may not be separated because the sequence may run like this. In particular for enzymes, the active site is very often not sequence consecutive. So when you look at a sequence, you typically don't have a motif that says enzyme is active site is here. Very often you have something, histidine triad is a very common example where you have a histidine that may be spaced here by 20 other or 30 other residues. And those 3 histidines are exactly what is making the binding site. You can imagine this somehow, okay, my finger analogy is not quite right. Now it becomes quite right. Something like this, okay? And those are the 3 histidines that somehow bind coordinated metal, and they have to have a certain proximity. Now in the sequence motif, you don't see that at all, and here may be some other binding motif, so you have 2 different AC numbers, in something that is completely together in sequence, right?
And in the case you have like an enzyme and it has a ligand that changes its active site, I mean one active site for 2 different functions, which could happen.
That does happen, yes.
What is that?
Actually, that's so typically people that is the argument why biochemists say this is not enough, the classification. Typically it carries the name of the one that is first discovered. Okay, this has all kinds of problems. We go back into that in more detail. This was just, I wanted to expose. I have several of these where I decided that I'd like to flash something at you and then repeat the same thing in more detail in 2 weeks from now or something like that, hoping that then at least it's sort of, there's a recognition event. Yeah? Here's another way in which we can do that, and that is called the gene ontology. It's just started by Michael Ashburner. And that, in fact, begins to have 3 separate, this in some sense is a forest, completely see 3 separate trees. One talks about the biological process. So the biological process, I briefly showed you an example for a cake pathway, where you have one protein does something to another protein, does something to another protein. At the end of it, you break down an amino acid, or you break down alcohol dehydrogenase. That's the one ADH I showed you that for. So essentially here, the aspect of function is simply what in the overall process does it have to do with degrading alcohol, for instance. And that's the process. Okay? The molecular function is really, this is the kinase or ligase that does a particular detailed biochemical reaction. The cellular component simply is where it is. Okay? Where exactly does that protein act? That is important because a lot of the interaction have to do with what you are next to. Okay? Interacting between two proteins happens sometimes because two proteins meet. And possibly if we would make other proteins meet, they would interact with those too. So some interactions are very specific, some are less specific. That's why localization is such an important issue. Here's an example for the gene ontology Go biological process for DNA metabolism. You see the typical tree. Now we actually have a couple of issues here. So here's the first thing, branching out. So you can come to this point here from two different parts. In the enzyme tree, nothing like that works. So the enzyme tree is a really strict tree. Here we have all kinds of cross connections, and in this classification, there is no equal depth. It completely depends on the Go number, how deep you can go into the tree. Sometimes it is a matter of more research will be done, and then the tree will be expanded. And sometimes really it is that for some types of proteins, there is not that amount of detailed subdivision possible. Okay, for some functions. But Go aspires, here's the biological process, here's the molecular function. So it's the same protein that it belongs to three different trees at the same time. It belongs to the process tree, it belongs to the molecular function tree, and it belongs to where I am tree, the cellular component, the localization tree. And in this tree, again, you can appear at different places. Here's the component, cell, cytoplasm, nucleus, nucleoplasm, and so forth and so on. So let's just begin with a couple of simple questions. Structure is conserved in sequence is a statement. So typically, similar sequence implies similar structure. That's this HSSP curve here, where you show number of residues aligned versus percentage sequence identity. So this is how many of the letters are identical. For those of you from the last semester, sorry that I show that again. And what you did for this, to derive this curve, is you had a bunch of proteins for which you know they have the same 3D structure, and a bunch of proteins in red, blue, in red where you know they do not have the same 3D structure, and you simply map them onto this graph, bless you, and you see that this black curve somehow separates these two sets of points in some way well. One way of saying it separates them well, and again this is sort of visually misleading, is there is not a lot of red above that curve. So whenever you get at this point, very few proteins above that curve that evolved do not have similar structure. Now this can be used to predict structure through comparative modeling, through homology modeling. So homology modeling, simply the idea is, and the same is for function, yes, for structure. The idea always is you have one protein, like the cytochrome in spearmint, you know what it does, you know how it looks. You have another one that is very similar in sequence. You simply assume that the two being similar in sequence do the same thing. You have the same structure, do the same thing. That's what you do for homology modeling. There you simply rebuild the three-dimensional structure from the three-dimensional structure that you experimentally know from the homolog, from the related one. And here, just a few exceptions. Not always the similar sequence is giving a similar function. Not always a similar function is coming from a similar sequence. So meaning two different proteins can do the same thing. And two very, very similar sequences can do very different things. Here's an example, an extreme example of that. And in some sense, this is even more extreme than the question that you asked, where we have the same protein, same sequence, same protein, and two different tissues doing slightly different functions. How could you imagine that? So you said about different binding sites. Actually, one idea you already had is that you sort of fill up one binding site and then then reuse it again. But this is not quite it, because then that would be the same in both tissues, right? So how could the function change?
Acidity.
Yeah, the environment. And acidity is just one example of an environment. In some sense, this idea about you sort of saturate one binding site and then you can do something else is another way of viewing it, so you could bind to it. And the binding to it could be specific in the tissue. How could it be specific to the tissue, what you bind to? Okay, let's go back. This is something that I have not discussed. We have about 20,000 proteins in human. Okay? We have different tissue. We have different types of cells in our body. Do they express the same proteins? So the same 20,000, are they active in every single cell? No. Not. Any idea? Anybody willing to guess how much is similar? Or similar is another point. Of these 20,000, how many do you believe are active in a typical cell? Actually active, what I mean is, are really expressed, do something, are there? 15%. This is a number that is much, much lower than I thought. No, it's more than that. In fact, I was surprised to see that's about a half only. And it's not, so when we look at the common subset that they all have, maybe then we get into the ballpark of your 15%. That's not far away from it. So when I'm looking at what essentially do all cells express, this is ballpark of 15%. But then they really have big differences, and overall, typically, a cell expresses in the ballpark of half to a little bit more of all the proteins. Ballpark, right? The amount of protein, so I'm only asking one of these 20,000, I get a 1 or 0 for them, right? Expressed or not. The amount of protein that is expressed is also very different. In some tissue types, some cells are way more expressed than others. In fact, it is by far not true that each protein is sort of expressed once. Some proteins are highly overexpressed, for instance, heat shock proteins. This is one of the background issues here. Here is an example. It's difficult to see, but I will show this again in a way that is a little bit better to see. So you have two proteins that essentially do the same thing, and although you are not that used to looking at structures, what I'm looking at on top is the binding site. I believe what you can clearly agree with me is that left and right does not look completely alike. Okay? So it's absolutely clear this is not just exactly the same way of doing it. I apologize that it's a little bit subtle, but you do see that the way the binding site is embedded here, it really is actually very different. So you can sometimes really do, in a more extreme case, next week, you can really do sometimes the same function with very different scaffold. The domain issue is simply, well, we have a protein that is, in this particular case here, 500 residues long. I look at another protein that I align, that I find some similarity in this red region here. And from this, I can infer that the red region is somehow similar in structure, similar in sequence, similar in function. So it is similar in sequence, that's what I see. And from that, I infer that it may be similar in structure, but I infer that it may be similar in function. Now, this one here may come with an annotation. But again, back to the same story with the enzymes, and most annotations, I do not exactly know where they come from. So I know the name here, that is an enzyme, ATP sulfurylase, sulfurylase, I'm not a biochemist. And here is the revenge of it. And that's a synthase. Now, what we do see, they're both enzymes, for those who can read those names. But again, the enzymatic activity may be related to that domain, and it may be related to that domain. So it could be that, in fact, this one here is doing something to ATP, and this one is also doing something to ATP in the same region, but the annotation of this one here does not contain the ATP binding because it is not known. And the synthase, uridine synthase activity is somewhere here. Okay? It could be that the ATP binding is here, and that this alignment is something else altogether different. Okay? That's the problem in the annotation of functions. So there are a couple of problems here. The first one is to really find what are the matching regions. The second one is to see what, from my original annotation, can I infer for the matching region. The complexity at which biology typically happens today is you read the name, you see the red, and that's the end of the story. That typically most of the time is what happens, because getting into much more detail requires a lot of computational biology and a lot of sequence analysis. Most people will not do that. Okay? So most people will not do what most of this lecture here is about. In this particular case here, it turns out that this is in fact a protein for which we don't know. Does anybody in the room who has not taken last semester's course know what a moonlighting protein might be? You know what moonlighting jobs are? No? So originally this comes from New York. So you have a job when the moon is up at night, and you have a job during the day, because one job is just not enough to pay the rent. So we begin to see this is a bunch of proteins that really have two different functions. Okay? And they're not of the type that you described, where essentially one binding site is covered and then that activates another binding site, because typically that's part of a process, the image that you haven't had in your head. This is typically really two completely different things, and two different involvements and two different processes. And again, the reason why this could be interesting is because it allows you to do more things with the same materials that you have. You have only 20,000 genes to express, and essentially you put the burden. So you have this thing that does two different things, and the burden about which one is done when is essentially put upon the environment, upon the control, what is brought next to it, right? The control mechanism. And that, in fact, is what we see happen in eukaryotes a lot. There are way more of these, and I believe with this one, I finished this part of the introduction. I finish that part of the introduction. I don't finish it. He's still not right. Okay. So general challenges. Let's again, homology-based inference is assuming that I infer something based on some evolutionary relation. One challenge I already told you, annotations may not completely tell you the region, and from the alignment, I'm not sure what the corresponding part is. Let's talk about something completely different, about evolution. So homology has to do with evolution. Let's just assume we have two different species, and here's a speciation event. Again, for people who have not taken last semester's course, what is a speciation event defined like in biology? Or not the event itself. Any idea how two species are defined?
We actually talked about it, I think, during the first or second lesson. It's when there's a physical separation between two groups of originally the same species.
Yes.
And they evolve in radically different environments.
Yes, so yes, that is very true. So the typical biological criterion, in fact, this is an important thing, because I have, for most of my life, I haven't thought about it that way. I always assumed that what you have is a genetic drift. So you have a subgroup, the subgroup has certain preferences for sun, and then they go to Spain or something like that, right? And the others stay in Munich. But it seems to be really much more this idea that suddenly disaster strikes. So that suddenly there's essentially a random subgroup, and the speciation event typically hits in a way where there is no genetic mark up. There's no group that has sort of a genotype affiliation toward there. Typically it is some outside event that separates the group, and that then in the aftermath of it creates maybe the fondness for sun or fun or whatever. But back to the speciation, or the species definition in biology, typically is that the offspring from two species can mate, but will not have offspring. Okay? So the fertility of the offspring. He also talked about the... question? No. I talked about the fact that we have Neanderthal gene in us, everybody in this room, as about 2% of the genome related to Neanderthal, and you immediately see there's a crack in the image, right? Neanderthal, Homo sapiens and Homo neanderthalensis are different species, and we carry Neanderthal genes, so the species definition doesn't always work. Okay, that's clear. But in principle, that is the species definition in biology, and that again is this idea: you have these two species, they are happily munching the banana, and then disaster strikes, the river comes, and then they are on different sides of this river, and ultimately they evolve different preferences for different food, because simply on the island you have coconut, on the other mainland you have banana, and at some point they come together, and whoever loves banana will not mate with the people who love coconut, or something like that. I don't know. Maybe for people it's not quite like that, but maybe for flies, in fact, you can do these experiments. It's pretty much like that. Here it's a little bit more modernized way of the banana eating. You can feed them differently, and you will avoid you can essentially induce speciation events. Sometimes the story is not as simple. So for these hooded crows, carrion, it's not in fact entirely clear whether it's the same species or not. Here, same species interbreed for some animals. So yes, it did happen in human, but for some species it happens more often. But still, now back again to our universe of protein sequences. So deriving the evolutionary tree may not always be easy. But on the level of the protein, where we infer homology, how can we get the tree there, right? And that, in fact, is even more complicated. But again, I said in this ADH, we have different ways to classify it. This would be a Wikipedia entry with all the database entries. And one of these entries here is the 1111. That's the enzyme classification number, right? There's an alcohol dehydrogenase. Here's the GO tree, one of the GO views of that. Now let's look at the assumed evolutionary relation here. So this may now be the ADH in human, this may be the ADH in plant, there may be some in yeast, and at some point there was one in bacteria. Now, we cannot really distinguish between this and this. What we see is those, right? We do not see these events. We infer these events. But we infer these events from whatever is left after a long evolution that mix and matches parts of the region. It's not somehow species survive as sort of entire things. Proteins don't. During the course of evolution, they exchange domains, they exchange pieces, they exchange bits and pieces, really. It's like a Lego box. You put a little bit of this, a little bit of that, and then it's very complicated. You can still say that these are related. May even be able to say that this, at some point, the common ancestor between these here and between all these three has a different time. You can say that too. You can say they are more similar to each other than they are to this one, which implies, if there is an evolutionary relation, it implies something about the distance of the common ancestor. You may not put a timeline on it, but you can easily put a relative time. This is later than this. That you can clearly do. But it could have been something like that. There could have been a common ancestor that came to all of them, and then they evolved differently, because in different species there's a different pressure. You know, viruses, HIV evolve very quickly, right? So this is very difficult to say, and it's also very difficult to say whether there was one common ancestor, or, back to your question, a forest of ancestors. That brings us to the terminology here of homology. So this may be another one here that is similar in sequence, and that may not have a common ancestor, for instance, and that is the issue about orthology and homology. The idea really is homology means that they're evolutionarily related. Orthology means they have similar function. Paralogy means they have evolved to a different function. So the idea really is, imagine I have this protein in a bacterium, and then I have sort of several bacteria that come off it, and I take the same protein in this bacterium, Don't I have that in my slides? No, I'm not really giving you this example. So I can do it on the whiteboard. So here we have the original one. Now assume in my new speciation event, because of some reason or other, I do a double. So this bacterium here, in fact, that is the ancestor of this one, has two versions of P. So that is P1, P2, it's P21, P22 or something like that. And we are here P23, P23, P3, 1, P3, 2, P3, 1A or something, 1A and B. So I have a duplication event. In the case of the duplication event, what happens is, say this protein does something that is absolutely essential. It digests alcohol, and that is very, very important for that bacterium to survive. It's its food source. But we have a copy that does it. This one tags along. So I have another copy. What can I do with the other copy then?
Remove it.
You could remove it. You could argue, in fact, this does happen. You could, so seen for me, thinking about simplicity in life, I would always say it costs energy to make this protein, so why not throw it away? Funny enough, biology does not do that. It is immensely, very often about a waste of energy from what I see, to gain some new opportunity. So why could it be around? Why could it help you to be around?
Maybe it's an adaptation of something. As in, I don't know, the cell, the environment has been changed, and maybe alcohol is not coming, for example, through cells, but through skin or something like this.
So you could put the one into the bottom, and one you keep where you have it, and the other one you put somewhere else. Is that what you're saying? Yes, that would be exactly one way of doing it. So.
Yeah, I was going to say, if there's one protein that mutates and that cannot digest alcohol anymore, we still have the other copy that can do it, so it improves survivability.
Why does the one that can no longer digest alcohol improve survivability, the ability to survive?
Because we have another copy which
yes, but so then we can remove it.
Yeah.
Maybe with that mutation you got an optimal function in some other environment, like.
So we have two. Now you two have two different ideas. So one is that you essentially have the same function, but you do it somewhere else. Essentially, you have a second copy that you use for a second purpose. Maybe put it somewhere else. Maybe just do it twice as fast or something like that. Another one is do the change and do something new with it. Exactly. So the idea is that, in fact, that's what people then would call the ortho log, that does sort of the same thing, and that they would call the paralog, and they would assume that this paralog is free to do something new. And free to do something new is, if you now look at P1 and you want to infer function, then this one here will no longer have the same function because it has mutated away. As long as you have identified that this is the ortholog, that is the one that does the same function, that as long as you would know that this bacterium really still needs alcohol. Okay, that's now the problem, of course, is to identify those. And sometimes the function can be driven by very few residues, so distinguishing one from the other may be a problem. The other problem really is the domain problem shown here. So this is the same story that I showed you before. You have this protein up here, you have a similarity in the red region here to this protein, and the function may be assigned to this one, this function may be assigned to this one. Now, in this particular case, actually, this assignment here, this is not consistent. But if you wouldn't have that one here, you wouldn't know. So if you only had these two, this is called function 1, you would infer that this is called function 1 too, because it has the same green region. These two have the same red region, so you would also give them the same function, but immediately you see that there's some clash here. But for you to see the clash, you either need to exactly know where this functional region is, or you need to have, in all cases, the one in between. And that, again, is like the ancestor. In many cases, we don't have the one in between. Moonlighting we talked about. Now, the first question is, how well does this homology inference work? So again, what homology inference is I start with a sequence here, and I align it against a family, in this particular case here, and whenever I hit on something that has an experimental annotation, I say, okay, this is my annotation that I will use for that protein up there. In this particular case, I make an example in which I want to infer that the protein up that I look with is a methyltransferase. This is the annotation of the protein that I happen to have used for this example, but assume we don't know that. Okay, assume that's exactly what we want to infer. So the first hit I find in the alignment here, the first protein in an alignment of similarity, and I sort by the first one is the most similar one, highest number of identical residues. That has an annotation. The first one that has an annotation but that is a methyltransferase. So I would infer this is a methyltransferase. Then I go further down in my threshold of what I allow, and I get to a certain level. I get another methyltransferase here at 66% sequence identity. At 66% sequence identity, I also find another non-methyltransferase. I'm not going to say it again. I stumbled over it the last time. So that is a mistake. 2 and 3 right, and in this particular case, that we don't know yet, the coverage, let's go on. If I went on, I would see that I find more wrong ones, but actually also more right ones. So now that brings us to the simple question. Should I use this threshold or that threshold?
How do you know if they are good or wrong?
So in this particular case, it's an example that I made up. In this particular case, I know for the one I start with what it is. And my goal is to infer that it is, in fact, a methyltransferase. I simply look up what I find. And if I had not known, what would it be? This is what I'm trying to do here. But now we have a simple case where you could say I find a threshold for making a decision in the future, right? And this is, in fact, my first part today is about adjusting thresholds such that you can do comparative modeling or homology inference of function. But the first question here is, which threshold would you use?
But I have a question. I mean, when would you use a threshold and not like keep the parts that are important? I mean, the parts that are the same and are methyltransferase domains. I mean, it's not. Yeah, I mean, it's not like.
Yeah. So in this particular case, you would write. So essentially, what you say, you're not getting out of the. Let me just follow up on your example. Let me first interpret what you say. What you said is, what I understood you said, is that forget about this threshold, methyltransferase. What I'm going to. I take whatever I see here on the whiteboard, and that is what we discuss in a minute. But I take whatever I see on the whiteboard, and I simply count how many methyltransferases do I find. How many formyltransferases I find, carbon myltransferase, amino, whatever. How many others we find, right? And then I count, I see this one is only once, this one is only once, this is twice, methyltransferase is 3 times, so I take a methyltransferase. Is that what you say?
No, what I was trying to say is if you, I mean, you could have a 99% match, but the function is not the same. Mm-hmm. So just keep the parts that are important for the function and say if this is...
do you mean use part of that word? Or what do you mean by part of the...
I mean, if you find the methyltransferase and then a 66% match, but it's still methyltransferase, and then a 61% match.
Yeah, but you don't know whether it's a methyltransferase. So, and again, in my example, I know that this first one is a methyltransferase. But in reality, I want a threshold without knowing this.
Okay.
I think what she's trying maybe to say, what I understood, is that do you know where the active sites are? No, so okay.
No, that's what I mean. That's what he just answered. If you knew, it's easier, but if you don't know.
I guess you would compare it to many other proteins as well and look for the most conserved.
But these are many others. So in this particular case, I'm a little bit slow in counting here, but I have 8 proteins that I find with annotations. So the alignment here is much larger than 8. So I should have explained it a little bit more in detail. I'm sorry. So I start with this protein here. I align against 50 million. Of these 50 million, I show some top hits part. In this top hits, I look up which one of those has an experimental annotation about what they do. Those I write. Again, it's a made-up example, but in principle, that's the procedure here, right? So that's what I get. For many proteins, I get the assignment of what they could do. So question which one is right. And the simplest question really is there a threshold that would help me to choose whether it's a methyltransferase or not? Dimitri, long due.
Shouldn't this threshold be a function of a sequence length? Because if a sequence
Yes. It should be, yes, we get back into that. It's a very good point, yes. But let's stay, before we get into that complication, the question to you is a much, much simpler one at the moment. I have two thresholds that I give you.
we train our system, and we guess a bunch of proteins we know, then we compare them to others.
That I will get in a minute. What did you say, the first one?
Yeah,
Why?
because we have more accuracy.
Lower
We
coverage,
have less coverage, but
which is more important. I get back to both of you. Let me still finish up on the algorithm that I misunderstood you to say. So if you counted, you group for majority, this is sort of chickening out because the whiteboard could be much longer if I just went to anything in the database, right? So at some point I need to put another threshold where I say, okay, this subset above that threshold I will count and it will sort of cluster if I wanted to do. So I clearly need some threshold just to motivate that. So ultimately, again, the question is 66% accuracy, 50% coverage better, or 38% accuracy and 100% coverage? And in fact, ultimately, the answer is not clear. It very much depends on who you tailor this method to, what they want to see, how they can handle that. There is no clear-cut answer. You could try to come up with a statistical way of gauging this. Say which one would, for instance, one important question here is, if you randomly toss something, what would be the answer to that? And clearly, if the accuracy level that you report is like random, then that is not the right threshold. It may still not decide between these two alternatives, but if one of them looks like random, then clearly that's the wrong threshold. Okay, this way it would work. It would not work more positively to say between two things that are not random, it does not give you a distinction. But again, problem specific, and for a variety of problems we will give answers that are specific to those problems. Let's look at another specific problem here. We infer subcellular localization. So subcellular localization, and that is work from Rajesh Nair, who is now at the Federal Drug Agency. This is a eukaryotic cell, or a sketch of the eukaryotic cell. Inside you see the nucleus, you see different organelles, so essentially you can imagine this like a bag of subcompartments. Subcompartments do different things. Again, here's the nucleus. This is a much simpler way of doing it, and we will, for this talk, I will even have a simpler one where I say essentially there's a nucleus, there's a cytoplasm, and there's an extracellular space, so there's an outside of the cell. And now my goal would be, can I find a threshold that would allow me to infer that proteins are in one of these compartments? How would I do that? Well, again, now ignoring the midnight type of moonlighting proteins, if two proteins are identical, essentially I would know they are typically in the same compartment. Again, there are exceptions, I say that. But then, the less sequence similarity they have, the less likely they will be in the same compartment. And somehow what we expect is some transition. In this particular case, we expect a transition that is nonlinear. So like a phase transition here, where we hit a zone, the twilight zone, so very much in the safe zone. We can infer that these two things have a similar, from the sequence identity, I can infer that they will most likely have similar subcellular organizations. There will be a region where I can absolutely not do that. And there will be some region where I sort of am right sometimes. Okay? And phase transition simply means that we don't have a linear point here. This is a rapid fall of the feature, and that is the twilight zone. So, in fact, if I wanted to know where this zone, where this threshold falls for the feature of inferring subcellular localization, how would I do that? So I want to, what I showed you before with the methyltransferase is one feature, methyltransferase, enzymatic activity. Now I'm trying to set up the same sort of threshold idea for some other feature, namely where it is, whether it's a nuclear protein or not. How would you set up the threshold? How would you test it? How would you build a system that can do that? How would you essentially test which threshold is good? Can I use sequence similarity 10? Can I do homology inference? So I have 2 proteins that have similar sequences. For one, I know the subset localization. Hence, I can assign it for the other one if it is sequence similar enough. What does it mean, sequence similar enough? That is the question I'm asking. And I am seeking a procedure by which I can answer that. How would we do that?
Train our algorithm.
Train.
Machine learning methods. Yes.
Before machine learning, yes, you can call that an SVM with one input unit, threshold, T, and optimize the T. Yes, you can do that. But much simpler. So my first question is, where do you get the data from? What is it that you need as data? If I gave that as an assignment to you in your exercises, what would you need?
A database of proteins that have this subcellular localization.
Okay, so let's say I gave you a database with 1,000 nuclear, 1,000 cytoplasmic, and 1,000 extracellular proteins. Okay? So my first question, are these numbers such that you feel, well, we can start? Or would you say, hey, this is so big, or this is too big for my computer, or too small for me? Any comment on this?
Depends on the quality of the annotations.
That is a very, very, very clever comment. So if they are all wrong, then 1 million is not enough. That's a very, very good point. I have never thought about this. You're totally right. Yes, that's certainly true.
Yeah, but that depends on your goal. As in, for example, if you have a certain protein of, for example, EXT, why would you need all the others?
Oh, so let's just assume that my task here is a sort of 3-class inference. We could repeat this in a two-state system, where I say extracellular, so outside of the cell, and everything in. So you say inside versus outside, okay? But we can do that. I just started with a three level.
You have 3 features which you need to predict, and I think
Let's first simplify that. I take your point. So we have 1,000 that are extracellular and 1,000 that are, or 2,000 that are not extracellular. So 2,000 inside, 1,000 outside. I repeat my question. Assume the quality is the typical quality of biological data. That means there are many mistakes in it, but it's the best I can get. And I believe it's good enough for me to start working on it.
Well, maybe you can separate it into smaller segments. For example, for each, take a third. So, for example, 1/3 of outside, 1/3 of inside.
Can we do half?
You can do half. You can
Good.
do like.
That's much easier for me. So I'm sorry.
And then,
Is that what you mean?
yeah. And then, for example, you have pretty much 2 groups, and you can, as Dimitri said, I think that's what your idea was, to actually train separately on two tuples, I would say. So, for example, first.
Good point. Good
first
point.
third.
So we essentially what you talk about is cross-validation.
Yes.
But before we talk about cross-validation, the data quality is a crucial thing, but there's something else.
it's how big the actual data that you want to predict is. Like if you have 50,000.
That's an interesting point. This is a very interesting point. But typically, for instance, in last semester when we talked about protein secondary structure prediction, we had a method that used 100 proteins and predicted for every 50 million in the universe, and could do that. Okay, I'm tweaking it because the secondary structure prediction is not one where you have one secondary structure for the entire protein. Protein typically has a few hundred residues, and you have essentially a method 100 times a few hundred, so several tens of thousands cases. But still, those were enough for the universe. In this particular case, in principle, those are not the number for human. That is actually for all eukaryotes, or ballpark figure. Is that enough? And there's another question that is important here to ask. You haven't asked that yet.
Homogeneity of the data. I don't know.
How much?
No, I was talking about this, how homogeneous are these proteins?
That's the important point.
Diversity.
So what I could have given you essentially one family with 1,000 proteins that are all the same. Now, you may think I wouldn't be that mean to you, or databases wouldn't be that mean to you. But the reality is biology is exactly that mean to you because the probability of finding related data is very high. Any idea why that would be the case?
Because if data is not related, then it would not be very similar.
That is a very true statement, but it is not answering my question.
Rephrase the question.
And the sun is bright today.
Yeah,
But both of these statements have a similar bearing on my question. So my question really is, why so it could be that 1,000 cases that I find in a data set are well spread from the universe of all things. But in biology, that is not the case.
That means it depends on the way you formulate your question. If you're looking for something very specific, you will find very specific things.
No, my question is, if I look at any database, I claim, any biological database, I claim I have a high bias in the sense that I find many things that are similar. Why?
Because I think biology will focus on few species, a particular species, rather than the whole universe.
That is part of the truth. But that still would have the too many hands. I cannot handle the moment. But the issue with that one is this still would have 6,000 yeast proteins, and that would at least be 6,000 different proteins in yeast, right? If that would be the explanation. So that would still be a separation.
Maybe due to evolution, because if evolving in completely different paths, so for example, you have an animal that eats grass, and then
She will do the same thing.
He will be doing the same thing. I think you gave an example about teeth formation last semester or something like this. So it's not good, like the shape of teeth literally means how the object is going to survive. So if it's effective eating grass with this particular teeth,
you will see the same thing again and again. This is a very interesting. But this is, yes, you're right, and this proves that you learned something important. But let's just say you have been just making the difference between I don't know, BMW and an Audi, and I'm talking about the difference between a car and a plane. So my question is on a much lower resolution than your answer. You're going much more in the detail. I'm looking for something much more simpler. I'm not entirely sure who was the next one. This is just the highest hand. Who believes he was the next one of the three of you? It's unfair. You cannot know because your eyes there don't work. Yeah.
So maybe somehow, I don't know, tied with the avalanche effect. If we already have enough proteins of a certain family, then we can easily create mutations for a new protein from the same family.
Easy is what? Yeah.
Because we are working with tools which predict based on the similarities.
Yes, similarities, sequence alignment, I would call it.
Yeah. You had another point?
No.
So this is clearly one thing. So one thing is that we are biased because it's simpler to do the same thing in a different organism, and it still gives me a paper. That's sort of, let's call it the social argument. In some sense, the bad argument. The completely understandable part, however, for instance, is you find in ADH in a bacterium, you find out what it does, and you actually want to understand a human disease. That will not be enough for you. You will really try to find the same thing. If you cannot study it in human, you will try to study it in worm, or in fly, or in plant. And in fact, in many cases, you will do all of these, or different people out there will do all of those in order to get at the human one. Okay, so there is, in fact, in some senses, back to what you said, so prove that the truth works the same way in different organisms is going to be new experimental data. Important new experimental data in order to know how the disease works in human. So if I put a mouse model of a disease, that will not be enough. Or a bacterial model. So I will try to get as close to human as I can. That's a very positive reason. And the other reasons that you mentioned about the alignment, the bias towards alignment, the bias by the method, is essentially where the light falls, I look. And I've looked more closely at things that are similar to what I've seen already. Okay? So that's why there is a lot of bias. This bias we have to remove. Okay? So you have to cluster. Whenever you get a biological data set, in fact, whenever you do machine learning, whenever you do anything, you have to sort of look at overlap of the data. And that's one of the major issues why data mining goes wrong, because it's complicated to do that. It's not, very often the problem is that people don't think about it. That is a problem that you can easily solve. Well, you can solve in principle. But sometimes it's really hard to establish rules. What does it mean there's too much bias in it? And in some sense, what we want to infer here is, what is the threshold for inferring extracellular versus not extracellular proteins? And in some sense, that threshold or that mechanism defines bias or too much bias for a machine learning method. So somehow it is a process that needs an iteration. Now assume these would be the two data sets that are already unbiased, yes?
I'm not really sure I understood that biased stuff.
So let's just assume, so this could be an ADH in human, and it could be an ADH in mouse, and it could be an ADH in rat, and horse, and you don't learn much about the threshold because they're all related. They all have the same localization. This is not the kind of example where essentially how much sequence similarity do you need, because you have essentially one class of protein, one functional type of protein. And the reason why you have all of them is because there is some biological reason that you want to study them in different organisms. Another reason could be that, in fact, it's easy to study that. Somebody else has already published that. You already know where to look. Much easier to find it, and you still get it in paper. That's what I call the social reason. Well, you need to get your thesis, right? The world is full of reasons like that. We all need to survive in some sense. And so there are different reasons why the database is biased. Does that settle your question? I assume. Now, back to the issue. So now I have 2,000 non-extracellular, 1,000 extracellular, and in fact, is essentially unbiased. This is sort of two clean sets. And I want to establish a threshold. So the way I do that is I simply run all against all. So I take my 3,000 proteins, and I do an alignment against all of those. Right? And then I evolve a threshold. Of this type here, this is for structure, and I'm looking for something like that. So I take all the proteins with known localization, I take a sequence unique subset out of it, and then I run against all. Here's a little bit of a problem. Sequence unique subset means essentially a subset of the big set, for which I have annotations, but it's such that it's not all ADHs in there. Okay? Okay, if I took out a sequence unique subset, why do I compare the sequence unique subset against the big biased data set? Why don't I do sequence unique against sequence unique? Okay, in this particular case, because I want to infer homology inference, and I want to infer exactly where the other things that are close in sequence have similarities. So if I did this against itself, I would find 0. Okay, I need to find something that has bias in it. There's some limit at which I can reduce the bias if I want to see how very sequence similar proteins have similar subcellular localization. I need to allow for some bias in my data set. And okay, so here is what happens if I did the same as before. I map it onto the same number of residues aligned, percentage sequence identity. Blue means proteins that do have the same subcellular localization. Red, no, that's structure. Here is subcellular localization. This is structure. Here's subcellular localization. Red means they do not have the same subcellular localization. Blue, they do. Now, you do see this sort of, okay, even if I switch off the, if you all come, I show you the screen. The screen is good. Yeah.
It's not that bad.
Yes, the point is, so what I mean is, so what you may appreciate here is there's some red here, but up here is blue. Here, well, this was visible to you before, that there's red here. What is less clear to you, and maybe you can see it now, maybe not, there are red points here. So you have examples where you have a lot of residues aligned, so you have a long alignment, essentially sort of like the entire protein, and they have a high level of sequence identity. In structure, you never have a red one here. For these subcellular localization, you do. And that means two things. One, it does sort of work, and it doesn't always work. It's not a surefire thing. So now we have, in fact, to take these red and blue and translate it into a probability. Okay, and that we can do in different ways. So this is an HSSP distance HVAL. So essentially now what I'm going to show you is distance, orthogonal distance from this gray. In fact, that is what Dimitri said before. Sequence identity would not be enough for a threshold. We would have to consider the length of the alignment. And by looking at the distance of this curve, rather than just looking at the distance from the vertical line here, a horizontal line, I'm sorry, I consider both. It's a two-dimensional feature. Here I look at an expectation value, and here I look only at sequence identity. And essentially, the two curves always mean the same. This one is accuracy, and this is coverage. So coverage essentially means how much of all the extracellular proteins do I identify? And at this threshold, this curve here means how many of those that I do identify are right. Okay? So just put an example here at an HSP distance of 0 where essentially structure is assigned right. You see that your performance is down to something like 70-something%. And the identification here is about 60%. So you find 60% of the secreted or extracellular proteins, and of all the proteins you find, about 70% are right. Okay? Roundabout. You can do it with this e-value. You can stay in this e-value. You don't find a better point in the e-value, and you find a much worse point. And this one here is what it says. But one thing is clear. None of these really work at 100%. So you cannot infer subcellular localization only by this. There are very red points up here. Why? Could you imagine anything that you already have learned in this course that would explain why? Well, it was sort of in passing. Not from me. Remember this talk from the IDP guy? He had the nuclear localization signal. Nuclear localization signal are very short stretches of highly positively charged residues. And you can essentially put them onto a piece of gold and put the gold into the nucleus. If the experiment has been done, right? So as long as you have this marker of 5 residues, you attach it to string it up to anything, that is put into the nucleus. And that means you can have 2 identical proteins with a tiny 5 residue difference. They would absolutely sit in this red region here. One would be nucleus, end up in the nucleus, and one not. Okay, so there's another mechanism. Overall sequence gives you a signal. It's enough to give a high probability assignment, but it's not enough to do it without mistakes, and it's just one simple feature. Okay, I believe with this we leave it here for today and get into the photo. But before we get into the photo, any questions? Photo. So photo, what I mean is I'd like a